With both fatty acids and ketone bodies, because they get converted into acetyl-coA, and that's how they enter the metabolic pathways at the level of the Krebs cycle. First of all, you're bypassing glycolysis and fermentation, so those pathways that we know are extremely upregulated in cancer and very important for cancer to have kind of optimal proliferative capacity or optimal growth capacity. So you're not supplying energy substrates for those important pathways for cancer. And then also, as you mentioned, there's no net carbon gain that can actually get converted into glucose. So fatty acids nor ketones are gluconeogenic precursors. So that's one idea of why maybe fatty acid metabolism, ketone metabolism is preferred in a tumor if you're trying to limit its growth.
Well, Angela, thanks for coming on. I'm really excited to be with you and kind of chat. We're talking offline a little bit about kind of the metabolic origin of cancer. And just as like a personal story, in the last year I've had 2 friends pass away in their 30s of cancer, and a family member got diagnosed with aggressive uterine cancer. And so it's kind of hit home for a lot of people, you know, the prevalence, and I think the 3rd leading cause of mortality, at least in the US, is cancer, right? And a lot of practitioners are not really addressing blood sugar dysregulation as a primary kind of phenotypic characteristic of cancer. And so I'd love for you to kind of give our listeners a little overview of how dysfunctional metabolic regulation contributes to cancer.
Okay, sure. When we think about the attributes of cancer, so what makes a cancerous cell cancer, we think of different hallmarks, so things that cell must be able to do, like supporting its proliferative signaling, so making sure that it keeps dividing, making sure that the cell will not respond to signals that tell it to initiate apoptosis or programmed cell death. And various other attributes. And these hallmarks have been recognized for a very long time, and it's always been the idea that these are conferred through genetic mutations that occur in the nucleus of the cancer cell. And that's absolutely true. The genetic mutations in the cancer nuclei do confer the behaviors of the cancer cell. But there's this reemerging, and I say reemerging because it was actually a very old idea, that perhaps in some or maybe even most cancers, these phenotypes are downstream of a metabolic problem. And when I say that, I'm referring to the metabolic pathways that regulate energy production and biomass synthesis in the cell. So basically what we know is that cancer cells have very dysregulated metabolism. So our healthy cells take in, kind of a normal amount of glucose and metabolize it through glycolysis and then convert it to pyruvate, and then that enters the mitochondria. And they utilize their mitochondria very efficiently to make the bulk of their ATP. And almost 100 years ago, a German biochemist named Otto Warburg showed that cancer cells don't undergo this normal type of energy metabolism. In fact, instead of shipping those energy substrates into the mitochondria to make ATP, you actually just have kind of a blunted path where you have lots of glucose uptake and then a lessened requirement of the mitochondria to make ATP. So essentially what you see in cancer, it's a really common phenotype across cancer types, is a very high upregulation of glucose intake and then lactate production instead of sending it to the mitochondria. And so this is called the Warburg Effect. And Otto Warburg first proposed, he said, well, and they do this regardless of if oxygen is present or not.
Which is really interesting.
Yes.
Okay.
Exactly.
Good point. Thanks.
Yeah, so usually in our normal cells, you only utilize that fermentation, the lactate fermentation, if oxygen is limited. So, but what the Warburg effect is saying that cancer cells do this even if oxygen is present. And so he proposed at that time, he hypothesized, he said, well, they must be doing this because their mitochondria are dysfunctional. So they have to make ATP a different way. And he even went a step further and said this has to be the initiating cause of cancer. Now, that was before we knew about all the genetic mutations in cancer. And so when we discovered these, or when it was discovered that the nuclear genome was chock full of these mutations, the whole idea of metabolism kind of being an initiating cause of cancer kind of fell by the wayside because, oh, well, it's clearly these mutations, that's the problem. But what we're finding now is this reemergence of this idea that perhaps actually upstream of all of those mutations is that metabolic dysfunction. And it makes a lot of sense because we see widespread mitochondrial abnormalities in cancer, and it's not clear to what degree the mitochondria are dysfunctional in all cancers, but they do express lots of mutations and dysfunction in their morphology and various abnormalities. And the idea is that when mitochondrial dysfunction occurs, you have a great increase in the amount of oxidative stress. So mitochondria make free radicals. And then those free radicals and that oxidative stress, coupled with an energy failure by the dysfunctional mitochondria, which kind of limits DNA repair mechanisms, is a prime environment for now the collection of all of these genetic mutations in the nuclear genome. And so that's the idea, that upstream of the genetic mutations, you have this insult to the mitochondria that produces free radicals, which then enhance the mutability of the genome and then the collection of nuclear genetic mutations that then confer the cancer phenotype. Wow. So that's the idea. It just kind of adds like another piece to the puzzle, I think.
That's really, so it's not just the mitochondrial DNA that's damaged, because we know that they're more susceptible, they don't have the histones and things of that sort, but it's really the nuclear DNA and really damaged from metabolic dysregulation, mitochondrial dysregulation, and leakage of like free radical species from the mitochondria into the very
Exactly. And the mitochondria are, as you said, very susceptible to damage because they don't have these same repair mechanisms. You know, they're also very close to the site of the free radical production, so that's being made in the inner mitochondrial wall or membrane in the electron transport chain. So the mitochondria themselves are very susceptible. But the mitochondria also, through maintaining energy production, preserve the integrity of the nuclear genome. If you have energy failure, DNA repair is a highly energetic process. If you have energy failure, you have failure of DNA repair. And then conjoin that with increased oxidative stress, and you could see how the genetic mutations could occur. And so that's just this hypothesis, and it is a controversial idea. A lot of people are talking about it and are interested in studying it, but it'll be really interesting to see kind of how it turns out.
Definitely. You know, that's really interesting. I want to create kind of some examples for folks listening that don't have a background in genetics and things, but if we travel to a third world country, Indonesia or Mexico or something along those lines, and like the infrastructure and the roads, if the economy is not really thriving and you don't reinvest in that, so what you're saying is like if there's not enough energy production, we can't invest cellular energy in the form of ATP to repair the DNA. And so that's, I think that's a new concept for a lot of people, that this DNA repair. And why is it important? Because we're getting these spontaneous mutations and we're relying upon like tumor suppressor proteins and DNA repair enzymes to prevent those mutations from causing DNA to read the wrong messages, right? And aberrant growth.
And every time your cell divides, it has to copy its DNA, and inevitably there will be mistakes that are made, but thankfully we have these repair mechanisms that go and survey, make sure that things were done properly, and then fix things that were mistakes, mutations that occurred. So if you have a failure of those systems, in part because of perhaps a deficiency in energy availability, then you could see why you might be able to collect all of these genetic mutations. So that's not, that's the theory, and there's definitely evidence to support that may be what's happening. It's not been directly proven in cancer initiation, but it's definitely a viable possibility.
Interesting. And I would love to dive deeper a little bit into HDACs, the histone deacetylase inhibitors, a little bit later and how they repair DNA and keep it kind of the histones compact. But I want to go back to kind of what you're talking about when you introduced the Warburg effect. And when people, not everyone again knows glycolysis, kind of the splitting of sugar and so forth, but so basically cancer cells are stuck in like an interval of sorts. So when people are doing like a burst, running upstairs, they're relying upon that anaerobic glycolysis, right? So they're splitting sugar and their legs hurt because lactate is building up. But if we're walking our dog for an hour or doing a hike, we're really utilizing oxygen to make cellular energy, right? So even though oxygen is available, cancer cells are like doing that interval burst and making that lactic acid. Now, I've heard there's some properties of lactic acid that is favorable for the cancer environment. Can you talk about that?
Absolutely, and I think it is very important to acknowledge that there is a lot of the cancer field views the aerobic glycolysis or the Warburg effect as a beneficial adaptation of tumors. So as opposed to this idea that it's actually the only, kind of a consequence of impaired mitochondrial function, that it's actually an adaptive mechanism, because there are many aspects of utilizing this pathway that actually promote tumor growth. And one you just mentioned is lactate. Lactate is an acid. It gets exported into the extracellular environment of the tumor, and it dramatically increases the invasive capacity of the tumor and the ability to metastasize. And so you do have a promoting effect just by making a lot of lactate. But then also, kind of the biggest idea of a benefit of this phenotype is the idea that if you're not taking those glucose carbons all the way into the Krebs cycle in the mitochondria, which end up you lose the carbons as CO2 when they're metabolized in the mitochondria. You are instead shunting them towards biomass. And when you think about a growing tumor, it's having to make lots of new nucleotides, lots of new lipids and proteins to build a new tumor. And so there's this idea is basically that you're conserving the carbon skeleton by not metabolizing it in the mitochondria and instead shunting it towards biomass synthesis to build new tumor.
Wow, that's really novel. I have not heard that before. That's really exciting. So instead of fully like oxidizing and breaking down those 6 carbons on glucose, we're saving those to make new parts of cancer cells.
Exactly. And that's inevitably happening, absolutely happening. Yeah.
So what happens then, I mean, we'll talk about all the good stuff with ketones and fat and so forth, but because when we eat fat, it doesn't really back convert that well efficiently into glucose via gluconeogenesis, so we're really fostering those carbons through the mitochondrial matrix.
That's kind of the idea with both fatty acids and ketone bodies. Because they get converted into acetyl-CoA, and that's how they enter the metabolic pathways at the level of the Krebs cycle. First of all, you're bypassing glycolysis and fermentation, so those pathways that we know are extremely upregulated in cancer and very important for cancer to have kind of optimal proliferative capacity or optimal growth capacity. So you're first of all restricting you're not supplying energy substrates for those important pathways for cancer. And then also, as you mentioned, there's no net carbon gain that can actually get converted into glucose. So fatty acids nor ketones are gluconeogenic precursors. And so you can't supply through kind of shunting around like you can with certain amino acids, some precursor that could then eventually be converted into glucose and then utilized in those important pathways.
Wow.
So that's one idea of why maybe fatty acid metabolism, ketone metabolism is preferred in a tumor if you're trying to limit its growth.
Yeah, very interesting. And some of your recent research has found that beta-hydroxybutyrate actually inhibits cancer cell growth. Is that one of the mechanisms? Because I know you've also looked at this signaling switch called HIF1 Alpha, Hypoxia-inducible factor 1 alpha. And we've kind of talked about that on the show a little bit, and that's this, maybe you can talk about how the role that HIF1 Alpha has, and I liken it to when people are sedentary, we kind of understand that we're not getting that blood perfusion and so forth. So where does oxygen concentration come into this whole equation?
Okay, so we'll take a step back and think about tumors as they grow. So they're growing under very chaotic and very abnormal kind of signaling properties. And so the tumors are growing under these deranged conditions, and in order to grow past a certain size, the tumor has to make its own new blood vessels. Okay, but all of these signals are so abnormal that the blood vessels they make are not fully mature or functional. And so what you get is this very abnormal and inadequate vasculature or blood vessel kind of network in the growing tumor. And of course, the function of blood vessels is to carry oxygen and blood and nutrients to the tissue. And so within this tumor, with these deficient blood vessels, you find areas within the tumor that are not able to receive adequate amounts of oxygen from the blood or other nutrients. And so we would call those hypoxic areas. So hypoxic meaning low oxygen. And that's just purely because oxygen can't get to that region of the tumor. And in classic cancer fashion, the tumor, you would think that would be bad for the tumor, right? Not supplying it with its nutrients. No, of course, classic cancer fashion. It benefits the tumor in many, many ways. And so you actually see one of the major ones, as you've mentioned, is activation of hypoxia-inducible factor. And this is a transcription factor that gets turned on by hypoxia, or when oxygen is low, it gets activated. And HIF1 will directly regulate about 60 genes expression, but pleiotropically it can influence hundreds of genes. And many of these are pro-survival, pro-proliferative, anti-apoptotic, increasing invasive capacity of the tumor, and also reprogramming metabolism to make it more glycolytic, because glycolysis doesn't require oxygen, and the initial kind of stimulus was that hypoxia. So we have tried to exploit this aspect of cancer, the hypoxia, with hyperbaric oxygen therapy. And this is just where, so right now we're sitting in Tampa, so we're at sea level, we're breathing the atmosphere, 21% oxygen. But in hyperbaric oxygen, you would be in a hyperbaric chamber where you can increase the pressure above atmospheric pressure, and you increase the amount of oxygen being breathed, so from 21% to 100%. And what that does is actually increases the amount of oxygen that dissolves in the plasma of the blood. And essentially now it can diffuse into the tumor and reoxygenate those hypoxic areas and theoretically shut off HIF signaling, kind of offset those tumor promoting aspects of hypoxia. And so that's something that we've been interested in studying. And we actually found a nice synergy of hyperbaric oxygen with ketosis, the ketogenic diet, but also exogenous ketone supplements that was more potent than either, kind of therapy on their own.
Very, very interesting. The researcher Rainier Straub, does that ring a bell? He's done a lot of research on HIF1 Alpha in the context of autoimmune disease.
Interesting.
And showing that that's like a favorable signature that promotes lupus and scleroderma and rheumatoid arthritis. So he finds high levels of HIF1 Alpha, at least at the genetic transcript. I can't remember if it's stable or not. But and we do see elevated levels of HIF1 Alpha in adipocytes in overweight individuals as well. And so I've been really fascinated by this whole story. And so when you were telling me offline about your research connection there, I thought that was really interesting to kind of bring up. And so by embarking on hyperbaric oxygen therapy, what we're doing is we're saturating the tissues with oxygen. And so that HIF1 alpha signals, and then the dimmer switch is turned off. So then we can kind of restore. That's what we're saying.
That's kind of the idea. Absolutely.
Very interesting. But also what you said about the cancer cells. So they're laying down blood vessels. So VEGF is one of the signaling switches. It's like fertilizer for blood vessels. But they're not really good. Did I hear that right?
Yeah, essentially, They're the signaling that is telling them to build new blood vessels is occurring because of oncogene and mutations and these signaling pathways that are not normal. And so the conditions in which the tumor is trying to build new blood vessels is very torturous in and of itself and very chaotic. And so you get these vessels that are built that are very inadequate. They have holes in them, shunts between the arteries and the veins, and they're not a mature vasculature, and that's what causes the hypoxia. And in some areas of the tumor, even anoxia, which means completely no oxygen, and that tumor will, even parts of the tumor will die there. And it's actually critically important in cancer therapy right now, because radiation therapy and actually a lot of chemotherapies work because of causing oxygen-free radical production. So the hypoxic areas where you don't have oxygen available, when you hit it with radiation therapy, it's not going to respond to that treatment because it doesn't have the substrate necessary to make the free radicals that kill the cancer. So it is absolutely limiting for many of our standard of care therapies. So re-oxygenating the tumor is an important, potential way to restore sensitivity to like radiation therapy.
Okay, maybe this is a topic for another discussion, but how does the radiation oxygenate tissues?
So the radiation doesn't oxygenate tissues. The radiation causes the formation of free radicals.
I see. So it raises the threshold a little bit.
Yeah, okay. But these are oxygen radicals. So you have to have oxygen present in order for the radiation to stimulate oxygen radical production. See, so if there's no oxygen present and then you apply radiation, well, it's not going to make the free radicals that will kill the tumor.
I see.
And so then that area escapes treatment. And the major problem is that area survives and now is able, after therapy is removed, to repopulate the tumor, and it is inevitably much more aggressive because the hypoxia promotes aggressiveness of the tumor. So that region of the tumor that's escaping treatment because it was hypoxic is now what's left over to make a new, much more aggressive tumor, and you see a lot of times the tumors come back with vengeance at that
like
point.
Superman tumors, because they could withstand.
Yeah, exactly.
Interesting. So if we take just a small sidestep and just talk practical levels here for helping to slow down the dimmer switch on hypoxia, this is why maybe exercise may be linked inversely with all sorts of different diseases, right? So oxygenating tissues and moving?
Potentially, yeah. I think possibly. You know, we're interested in could you use lower levels of oxygen? Because right now we've studied hyperbaric oxygen. You have to have a hyperbaric chamber, which, you know, that's difficult to implement. They're not found everywhere. Not all hospitals have them and things like that. But we wonder, could you do maybe just 100% oxygen at atmosphere? Because that could be easy to apply. Will that have any therapeutic effects? And the data is kind of mixed. There is some data that suggests possibly, but theoretically, your hemoglobin is saturated at atmospheric normobaric normoxia. So being at atmosphere, breathing atmospheric levels of oxygen. And so there's this idea that you need that hyperbaric aspect to actually physically deliver more oxygen to the tissues. So it's not really clear. It's a possibility, but it may require a stronger stimulus like hyperbaric pressure.
Right.
Yeah.
Especially if maybe someone has active cancer, for example, really like treatment.
Yeah,
okay.
Possibly.
Very, very interesting. Let's talk about one more kind of bad thing, if we will, the cell membrane. So we know that from Dr. Seyfried's work, I do recall reading in the book that cell membrane integrity, and there's some different, calcium, potassium channels and stuff. Have you looked at that? And because the reason why I'm asking, all your research and a lot of research in the ketogenic world shows that a diet high in fat kind of helps cell membrane fluidity and EPA, DHA, and so forth. So
yeah, it's not something that we've looked at specifically. We do know that specifically mitochondrial membrane integrity is really important. If we're thinking about trying to kind of improve the health of our mitochondria as a potential tumor suppressor. So there's definitely a lot of data that show that healthy functioning mitochondria act as tumor suppressors. Wow. And Dr. Seyfried has written about this in his work, and that if you have, even there are some studies that have transplanted mitochondria of healthy cells into tumor cells, and you get a suppression of the tumor phenotype. And so anything that could kind of preserve mitochondrial membrane integrity, which then preserves mitochondrial function or helps preserve it, could be beneficial. This may be a reason that certain diets, like the ketogenic diet, might be having an effect like that. We've never worked on that specifically. I do think, you kind of mentioned EPA, DHA. I think that aspect, the specific fatty acid composition of the diet and how that influences the composition of our cell membranes, the mitochondrial membranes, and their functionality is very important. Yeah. And that's an aspect of ketogenic diet research I don't think we've, as a whole, gotten into too far yet, because right now we're still trying to, okay, does the ketogenic diet work, And then from there we need to really optimize the diet, because undoubtedly you could have a very poorly formulated ketogenic diet, which could have some bad effects, So making sure that the actual composition of the fats in the diet are the ones you want to be there is critical. So
really important point. I do see a lot of that, like on Instagram, people post like the food porn and food pictures, and a lot of it is like bacon and cheese, and it is low carb, but not really, like you mentioned, the type of fatty acids that you want to be getting a lot of.
Yeah, it's important to make sure that it's a well-formulated ketogenic diet.
Yeah, and maybe we can finish up with that towards the end. I want to get your perspective on that. But I think we've kind of tackled most of the deleterious effects and kind of what happens in the metabolic signature, if you will, of a aberrant cell. But one thing that we didn't really talk about as of yet is insulin. And we know that insulin kind of promotes growth and things of that along those lines, and then we also know that the ketogenic diet really kind of keeps that, kind of suppresses that insulin signaling a little bit. So have you looked at insulin much?
We have not specifically looked at insulin and insulin signaling, but it is absolutely recognized as a really potent growth factor in cancers, and many cancer types kind of, upregulate insulin receptors, IGF receptors. We know that insulin promotes tumor growth, and of course we know that in a whole system, what causes your insulin to spike mostly, is dietary carbohydrate. And so knowing that hyperglycemia and hyperinsulinemia, so high glucose and high insulin, are both strong promoters of cancer growth, it becomes kind of a obvious idea to try to suppress that. And diet is a great way to do that. And that's probably one of the major mechanisms of the ketogenic diet. And, on the ketogenic diet, you don't always get a really remarkable drop in your circulating blood glucose. But what I think is maybe more important is you don't get these spikes in carbohydrate meals that cause spikes in insulin. So you kind of have a plateau of glucose and insulin over the course of the day, or the time that you're eating the diet. And I think possibly what we're seeing is, rather than an absolute kind of like restriction of glucose from the tumor that's induced by the diet, I think it is restricted to some degree, mostly. There's some data that shows that. But I think it might be even as important, or maybe even more important, that you're blocking those spikes in insulin that potently drive tumor growth.
Very interesting.
So I think that's an aspect of it.
Yeah, just as a personal note, I've been tracking breath ketones and glucose for like a year and a half now, and I have noticed that blood glucose, it's not totally suppressed, right? I mean, sometimes it's 95 after a meal or 110 or whatever, but we don't see that huge, trough valley kind of relationship is what you're referring to.
Yeah.
Very interesting. As you were saying, sorry, Angela, I want to go back a little bit. It's kind of off topic, but the notion of not taking antioxidants when you have cancer, a lot of oncologists, and kind of, I know we're not giving medical advice here, just kind of talking about mechanisms. But I remember from one of the videos I was watching that a talk that you gave online, that cancers love, and we talked a little bit, love oxidative stress. And so the idea is that when you take an antioxidant, you kind of maybe even empower the cancer cell a little bit?
It's very possible. This is a really interesting idea that people are investigating right now. And oxidative stress is really a double-edged sword for cancer. So cancer cells at a basal level operate under more free radicals, more oxidative stress than our healthy tissues. And they benefit from that. So it stimulates growth factor signaling, it stimulates, kind of aggressiveness of the tumor. It can aid in the acquisition of new genetic mutations in the tumor, which help promote it. However, because they exist in this kind of heightened level of oxidative stress, it's thought that they are selectively vulnerable to further increasing oxidative stress, because they're closer to kind of this threshold limit. And above some threshold, any cell that experiences a certain amount of oxidative stress, it'll kill the cell. So the idea is they're kind of like existing, they're trying to get the best of both worlds. They have more oxidative stress and that benefits them, but we can manipulate that by providing pro-oxidant therapies to push them over that threshold. And like I mentioned, that's what radiation therapy does. That's also, many chemotherapies are known to work in part, at least, by this initiation of oxidative stress. So the idea is, if you're receiving a pro-oxidant therapy for a treatment, but you're taking antioxidants, are you undermining the efficacy of the therapy? You could be. It's possible. And there's definitely some data that shows that might be the case. But like I said, it's a very not well understood phenomenon, and it's very complicated because, for example, we saw a synergistic effect with the ketogenic diet, which is antioxidant in its effects, and hyperbaric oxygen, which is pro-oxidant in its effects. Dr. Adrian Scheck at the Barrow Neurological Institute has seen a very nice synergistic effect with the ketogenic diet and radiation therapy.
Interesting.
So
that would be in conflict with kind of that old hypothesis that...
So it's hard to say. There's definitely data suggesting both could be true. So I don't think we know yet.
Yeah. Okay. I just wanted to throw that in there. I'd never heard it articulated in such a way. So thanks for breaking it down.
Sure.
I think that gives people a better understanding. Because I think people think, oh, my doctor or my oncologist just doesn't understand the power of antioxidants, and they're just telling me this. But there's a real logical reason as to why that is. Okay, so let's talk specifically about some of your research and the exciting things. And what we were talking offline a little bit, and you're like excited about beta-hydroxybutyrate and these other ketones and how they're pleiotropic. They wear a lot of hats. They do a lot of things. So it's not one mechanism. So we see in traditional allopathic medicine, it's single drug, single target. That's the approach. But what's so unique about the state of ketosis is it's like this network that's being upregulated. So what are some of the hot points that folks should know about in terms of being in a state of ketosis?
So there are a number of effects of ketosis that you can see why they could be anti-cancerous. So of course, just ketosis in general, where you have the lowering of glucose and insulin. And that was, of course, the original idea. The original idea never predicted that ketones would have anti-cancer effects, right? The idea was, oh, we know tumors suck up a lot of glucose. Well, let's try to limit glucose. The ketogenic diet does that. But now studies are showing, wait, the elevation in ketones is probably as important. And so what we're now seeing, it's been known for decades that ketones were energy metabolites. But really within the past 5 to 10 years, we're starting to see that they have these very important signaling and functional roles in the cells that have nothing to do with their role as energy metabolites. They influence gene expression. So they're histone deacetylase inhibitors, which means that they alter what genes are being expressed in the cell. Histone deacetylase inhibitors are an entire class of anti-cancer drugs. So we have pharmaceutical companies trying to develop these synthetic HDAC inhibitors as anti-cancer medicines. And beta-hydroxybutyrate a couple years ago was published, and it was discovered that it is an endogenous, our body makes it, and it's one of its functions is to inhibit class 1 and class 2 HDACs.
Which is so exciting.
Yeah, very
that's
cool.
What got me excited about this whole ketogenic story.
Exactly. And it's really neat because there have been studies that show that the ketogenic diet influences gene expression in the tumor. And it suggests that the diet kind of normalizes gene expression, makes it look more like the healthy tissue gene expression. And this could be why. This is now a new link that says, oh, well, it could be acting through and is this HDAC inhibition function, potentially to be restoring normal gene expression in the tumor. So that's one. Inflammation. Inflammation is a huge component in cancer origination and progression. And ketosis is anti-inflammatory. That's been known for a long time, kind of on a systemic level. You notice reductions in circulating levels of inflammatory cytokines when someone's eating a ketogenic diet. But more specifically, a couple of years ago, maybe 2 years ago now, beta-hydroxybutyrate was shown to inhibit the assembly of the NLRP3 inflammasome. So this is a component of the immune system that's activated during cancer, during aging, during lots of chronic illnesses that incites inflammation. And now BHB as a signaling molecule inhibits that formation of that inflammasome. And so it's again another potential link that explains maybe why ketones have these anti-cancer effects.
Can we pause? Sorry to interrupt you, but I just want to pause because a lot of our listeners have heard about NF-kappa B, right? It being like the cellular mediator that when activated from poor lifestyle choices, fast food, goes to the DNA, turns on bad genes that make, like you mentioned, like TNF-alpha and interleukin-6. Where in this cascade is the NLRP3 inflammasome? Is it outside of the cell? Like, so people have a better context of what it's doing.
Yeah, I am not... this is not an area that I study, so I don't want to say something inaccurately. I don't know if it is specifically in one location or the other. Yeah.
It's this big kind of network, but does it
but
correlate with
the Yeah, it's definitely part of the inflammatory process.
Sure.
And it produces many cytokines which then, you know, further activate other immune cells to initiate inflammatory signaling. So it's a component, it's a major component of the inflammatory response. I'm not sure specifically how it intersects with NF-kappa B.
Sure. Okay. I've had some questions about that. Some people were kind of curious, like, you know, they geek out.
That's a great question. Now I'm going to go look that up.
Yeah, I was trying to figure it out, you know, but if you read research in asthma or autoimmunity or intestinal permeability, they all refer, I shouldn't say they all, the scientists refer to suppression of the NLRP3 inflammasome. So it's a key mediator, and just to reinforce what you said, one of the main ketone bodies in the serum, beta-hydroxybutyrate, suppresses that. So that's a direct anti-inflammatory effect, which is really exciting and awesome.
Yeah, exactly.
So we have the kind of contraction of the histones, protecting our DNA, and then having other effects via the HDAC inhibition, suppressing NLRP3, which is awesome. Any other hot signaling?
Yeah, just to clarify quickly, so the histone deacetylase inhibitors actually end up opening up expression of certain genes. So you have like increased loosening of histones in specific regions that increase expression of those genes. So things like antioxidants is generally what was described when this role of BHB was discovered, but it's probably happening in other genes as well.
Okay.
So other things that could be having an effect, alterations in oxidative stress. So it's known that ketone metabolism, BHB metabolism, decreases free radical production. So you get less oxidative stress, and you have this increased antioxidant capacity. And as we already mentioned, you know, oxidative stress promotes the cancer phenotype.
Yeah.
So that could potentially be one effect. I have idea, I'm not sure that there's really any data that has tested this yet, but so ketones are taken into the cell by these same transporters that export lactate. I'm interested if that has some kind of inhibition on the ability of the cancer cell to get rid of its lactate and maybe reduce lactate availability in the extracellular environment. So that's another kind of potential mechanism. Ketones, there's been some data that suggests ketones can inhibit glycolysis. And again, glycolysis is of course a really important pathway in cancer. Ketones are very anti-catabolic in a catabolic state. So this is important to our evolution, kind of serving as a fat-derived energy substrate and then suppressing muscle breakdown, right, during starvation. And so ketosis in general, slows lean body mass loss. And that's really important for a cancer patient because cachexia, which is the loss of, especially of lean body mass, which is some of the most important and consequential effects of cachexia, is very damaging to the health of cancer patients. And so being in a ketotic state could potentially slow that. And there is some data that shows that the ketogenic diet slows cachexia and simultaneously decreases tumor growth. So decreases tumor growth and then prevents the body's muscles from wasting. So
really awesome.
Yeah, it's very interesting. There's a whole lot of potential mechanisms that we're interested in studying, but we just need more data. More data,
more time. Yeah, I mean, I commend you for all the work that you're doing because you're really helping us to understand, how diet kind of translates into health and disease.
Sure.
But it's interesting that you're kind of finishing up on that anti-catabolic effect, which is really unique because a lot of people want to preserve muscle throughout the lifespan. We know that higher levels of lean muscle mass really affect quality of life as folks get older and so forth. And I think a lot of people have this notion of the old school bodybuilding diet, really high protein, high carbohydrates, and eat 6 to 8 meals a day. And now what we're seeing is some people, Dominic and probably yourself, are having 1 or 2 meals a day and still maintaining that level of lean muscle mass. So Justin, I know you're fired up on that. I was actually at a gym this morning and met kind of a famous icon in the fitness space, a really big gentleman. And I was telling him about our interview and he was saying like, yeah, that's one of the reasons, like I love the ketogenic diet for health, but it's really hard to maintain lean muscle mass. And I didn't want to school him on this new research, but it's kind of fascinating. So Is that through mTOR signaling or elevated blood level leucine?
It's not clear,
Okay.
I don't think, and it's not my specific area, so I might just not be as knowledgeable on that as well. But I know there have been, actually within the past year, there was a novel mechanism of acetoacetate actually stimulating muscle protein synthesis, and this wasn't through mTOR, so it was a different, I think it was through the MEK-ERK pathway. I'm not positive, I'd have to double check that, but so this was just described within the last year, this new mechanism by which ketones can not just, that was actually promoting muscle protein synthesis, so which is perhaps different than slowing catabolism. So there seems to be just different methods and ways, and I think kind of the overall idea is that basically ketones, and how this affected us evolutionarily was that ketones provided a fuel to the brain, so you didn't have to break down your protein to give gluconeogenic amino acids so that you could make glucose for the brain under starvation conditions. So just in general, the state is very anti-catabolic, and so perhaps it's not surprising that we're starting to see specific molecular mechanisms that also confer that.
Yeah.
So
very interesting. Going back to that transporter protein that you mentioned, or cellular transport, where beta hydroxybutyrate goes into the cell and then also lactate goes out via that.
Yeah.
Is that a carnitine related structure?
No, that is the monocarboxylate transporters. So they transport the monocarboxylates, including like beta hydroxybutyrate, pyruvate, actually, lactate. There's a couple of different metabolites that go in and out through those transporters. So
interesting. Okay. So I think, Angela, we've covered a lot of bases. Anything about the ketogenic diet or your research or something that you just want people to know about that we didn't get to cover?
One exciting area that we mentioned previously off camera was some new data coming out of Dr. Adrienne Scheck's lab, and she's really a pioneer in the field. She's doing some fantastic work helping us understand mechanisms, because as we mentioned, there's so many with the ketogenic diet that, and mechanisms are important, especially in our field. People want to know how something's working, and that's an important question. Anyway, she's doing some great work, but she published earlier this year showing that the ketogenic diet actually kind of restores anti-tumor immunity. So increases our immune cells' ability to attack the cancer. So cancers, as they form and grow, find ways to evade immune detection. And essentially what she found was reduction in immunosuppressive signals. So the tumor will actually kind of like express certain signals that say, oh, immune system, don't come, neutralize me, essentially. And so there was a reduction in that ability, and because of this, the immune system was actually able to go and attack the tumor better than without, than not in the context of the ketogenic diet. And so I think that's fascinating. A lot of people are interested in immunotherapies in cancer right now, and enhancing. The immune system has the ability to neutralize and detect cancer. We've probably had cancer, small cancer cells, multiple times in our lives, but they've been properly detected and neutralized by the immune system, and tumors will only become a problem when that ability is lost. So if this diet is also in another way, aiding in our own immune system's ability to fight cancer, that's fantastic. So that's a cool area of research.
Really interesting. So is it that the cell's sending out like a different sonar message, or is it that the immune system just recognizes and seeks and destroys better?
So the immune cells express certain cellular markers, and so do the cancer cells, that depending on exactly what they are, it will either cause the immune cell to attack it or not, right? So it's just kind of an alteration in those, what's being expressed on the cell surface of the cancer cells and the immune cells that aid in that ability to detect and then neutralize.
That's really fascinating.
It's pretty cool.
Yeah. Awesome, Angela. Great stuff here. So let's kind of finish off a little bit about anything that you've learned over the years in the D'Agostino lab, and also working with a lot of patients with chronic disease. I know you go to a lot of seminars and interact with people about adherence or application to the ketogenic diet. Any tips or tricks for listeners?
So I do think it's important, especially in cancer. This is there's a fair amount of animal data out there, but there's not a lot of human data yet. And thankfully, you know, we are learning more, and I think there's at least 12, I think, clinical trials right now registered to look at the ketogenic diet and cancer. So, you know, I think it's important to keep in mind that, you know, everything we're talking about is in preclinical models and it has not been proven in humans, but it's very exciting. I think that in our field, it is important for us as researchers in ketogenic diet to learn from those that are studying the ketogenic diet in other fields. We have benefited so much from the information that has been put out over the past 100 years by the neuroscientists and the neurologists who have studied the ketogenic diet for epilepsy. So a lot of, you know, mechanistic work and just understanding application of the ketogenic diet, we have been able to really, you know, ride on their coattails, and that's amazing. And so continuing to look outside our own kind of narrow focus. Maybe I'm focused primarily on cancer, but there's a lot of information out there being discovered in neurological diseases or, you know, other metabolic type diseases where you can really gain a lot of information.
Sure.
And just integrating that into what, you know, we're doing. And I think that's a really important part kind of progressing the field. It's communication and translatability and interaction with other people in the field.
That's really important point. Kind of related to that, that's really why Silicon Valley is what it is. So a lot of people in tech kind of cross-pollinated at different companies, and so that collective really grew, whereas a lot of the tech companies back in the 1970s were actually in Boston, and it was really vertically integrated. Like you stayed within your company, you had a non-compete, so you know, I don't know if it's IBM or other, but they wouldn't cross-talk, whereas Silicon Valley, people go to the bars and communicate, and you got programmers talking with coders and all that. So
that's a great analogy. I
like that you're, you know, talking to neurologists in this, because specialists tend to get siloed in their little bubbles and look at, not outside their box. So
exactly.
Really awesome. So Angela, we want to get to know you on a personal level a little bit more. You know, you're busy, you're married and so forth. What does your morning look like? We know that really successful people kind of have a morning routine. Can you share with us yours?
My morning routine. I like to wake up early, spend a little bit of time with I have 2 dogs, 2 cats, and a husband. So enjoy having a morning coffee at home. A lot of times I try to work out in the mornings. I find that just sets me off on a better day, you know, just feeling so much better when I get out and get active first thing in the morning. So whether going to the gym or, you know, maybe a walk, we have a park right behind our house. Our dogs love to go out there and throw the frisbee, and that's always really nice. And then just most days, you know, heading into the lab and getting started on things. I don't know that I have, you know, a particularly interesting morning routine, but I definitely need family time, maybe some exercise, and a cup of coffee.
Are you doing fatty coffee or just straight?
I usually do MCT oil in my coffee, and or coconut oil sometimes, or sometimes butter. I kind of switch it up, but typically like a fatty coffee.
Yeah, and then exogenous ketones, do you take those supplementing?
I do some. I kind of do it as a as needed basis. I do try to do like intermittent fasting a few days a week where maybe I won't eat until the afternoon, you know, aside from my kind of buttery coffee or something. So sometimes I'll take, you know, exogenous ketone supplement, you know, if I start to get hungry around lunchtime, and generally that kind of satiates that and gives me some energy to kind of work through.
To push through that fast.
Yeah.
I see. Yeah. Okay. Any success stories about the exogenous ketones that you want to share? I know a lot of people have used them for various applications, but
it's a really interesting area of research, and it's really new, right? So, and it's coming off of all of these discoveries showing the direct benefits of ketones, and that's actually a big part of what our lab is interested in. Would it be possible to take, you know, an exogenous ketone supplement and get a similar level of therapeutic benefit as the ketogenic diet, or as good, and it probably depends on what condition you're looking at or what, you know, purpose you're looking at. And so of course, you know, we are just studying, you know, in preclinical disease states, but we have seen exogenous ketones have had a therapeutic effect in a wide variety of seizure models, which is really exciting. In my cancer model, which is a model of metastatic cancer, the exogenous ketones, which was a ketone ester that we were studying
Can you
at that
clarify
time.
how that's different?
Oh, sure,
Yeah.
sure. So there are various, anyways, like exogenous ketone is basically something that you consume that either already is a ketone or gets converted into ketones in your body or releases ketones. Okay, so you can create various, compounds that would, do that. So there are some like ketone salts, which are kind of simple compounds which directly kind of release ketones into the blood. Ketone esters are named such because they have an ester bond in them. And the one we utilize in the lab is, it basically contains acetoacetate, and then it contains a molecule that goes to the liver and gets converted to beta-hydroxybutyrate. So when you consume this ketone ester, you get an elevation in both of the primary ketone bodies, beta-hydroxybutyrate and acetoacetate. And that's the one we've done most of our work on in disease states. It's very potent, so it can induce a level of ketosis quite quickly and with a small dose. And that ester in my cancer model that I use actually had a very nice therapeutic effect. It was actually as effective as the ketogenic diet in my model.
Wow.
I don't know if that would be applicable to other cancer models or cancer types or in humans. We can't say, but that was very encouraging. So
awesome.
We're interested in furthering our understanding and the use and application of exogenous ketones, but we have a lot of work to do.
Yeah, I think it's really exciting. I personally think it really helps people understand how they will feel neurologically, and like you said, the food suppression, and kind of get the buy-in for them to start restricting their diet. But I see, kind of like, I don't want it to be used as like a cheat pill, like, oh, I'm gonna go have my McDonald's and it's gonna be like washed out with the ketones. And so I'm sure that's gonna invariably happen, right? People like the shortcuts. But so along those lines, you're stranded on a desert island, vitamin D and omega-3s are covered. What ketogenic food are you gonna bring with you?
What? Wow. Ketogenic food. Let's see. Probably coconut oil. I utilize it a lot in my daily life. I like the taste of it. It's an easy way to elevate your blood ketones naturally. You can do a lot of things with it. That's kind of my staple ketogenic food that I try to implement in my diet.
It's available anywhere too, which is great.
Exactly. Yeah. There's probably even coconuts on that island.
Right, you may not even need it. Get the real deal. That's awesome.
Yeah.
So as a final question here, if you were to bump shoulders with someone from the World Health Organization, maybe president of the US or prime minister of another country in an elevator, and they ask you about the ketogenic diet, what would you want them to know about it?
I would want them to know that the quality of the preclinical evidence that's coming out of the labs right now is so encouraging that we desperately need more funding to study this. And just the nature of funding mechanisms, funding has gone down over the past several years, which is making it ever more difficult for scientists to advance their work. And because the nature of ketogenic diet research makes it so that's a lot of the times not something that is going to get funded through traditional mechanisms, and that makes it really hard to further understand what place this actually has. And I would just want them to know that there is really, really encouraging evidence in so many different conditions. We study just in this lab, various neurological conditions, seizure disorders and cancer, and, people are investigating it in all kinds of different areas. So I would just want them to know that it deserves more attention and more funding than it's currently getting.
Right, and so to look at the flip side, like in the drug model, due to the fact that you can patent something and have like exclusivity, they can invest a lot of money in research, knowing that they're going to recuperate that down the road. So
and it all makes complete sense. Like I don't prescribe to the idea that there's some kind of conspiracy theory of people trying to suppress it. It's just realistically, research is expensive. Human trials are very expensive. Even a phase 1 trial is a minimum of $1 million probably. And so if you're talking about something that's not patentable, it's hard to raise the money to get something because there is no real return on the investment. So, but despite that, even since I started getting in this field, I've seen such extreme interest build, from the top layers of, the scientific organizations in this country. So I'm very encouraged. You always want it to be a little faster than it is.
Yeah,
but
it's I think just like on a layman perspective, it's growing so rapidly. I mean, it's amazing. Yeah. It's like a new religion. People are just die hard, and it's cool to see. So I think we're going to see like a new revolution from the ground up kind of thing, like pushing scientific research, hopefully. So, Angela, I really appreciate you coming on the show again. Commend you for all your great work. If folks want to learn more about your work, the D'Agostino Lab, where can they connect with you online?
Yeah, so I have a Facebook page, Angela Poff PhD on Facebook. There I try to post, updates on research that's coming out there, some of the work that we do. Would probably be a good spot. Dr. D'Agostino runs a website called ketonutrition.org, and he posts a lot of kind of what we were talking about and other good information and work coming out of our lab, and that's always a good option as well.
Awesome.
Yeah.
All right, folks, thanks so much for tuning in. I'll put the show notes and the links below this video associated with this video, and if you liked it, please give it a thumbs up and subscribe to the channel. So, Angela, thanks again for coming on.
Thanks, Mike. Really appreciate it.